All right. Well, good morning, good afternoon, good evening to all of our ISCT members joining us around the world. Welcome to our first telegraphed featured member segment of 2023. We are starting this year strong with a conversation with the distinguished Dr. Bernd Giebel. Dr. Giebel runs a research group within the Institute of Transfusion Medicine at the University Hospital Essen in Essen, Germany. His exemplary research career has taken him all the way from hematopoietic stem and progenitor cells to cutting edge work with exosomes and specifically exosomes derived from mesenchymal stromal cells, which I'm excited to discuss in greater detail with you. And so with that, thank you, Bernd, for taking the time to chat with me today.
Yeah, thank you, Ashley, and hello, everybody. So I'm happy to be the first interviewed partner in 2023, and I'm looking forward for a good chat.
Great. Well, just to start off, can you tell people how long you've been a member of ISCT, how you got involved with the society?
Yeah, metric data. So I think I became exosome co-chair in 2018, and it might also be the time I stepped in. But I had been attending some of the meetings before, then with the interest of the stem cell research. But yeah, I was maybe actively recruited and asked whether I'd like to set up the exosome committee together with Psyche Lim.
Oh, great. Do you remember what your first ISCT meeting was?
Um, one was in Singapore quite early, and of course, they was extremely excited, and so yeah, it was quite warm outside, and then with the air condition, it was a change, and but it was a great place, and I really appreciated it.
Oh, that's great. And like you said, I mean, you've been in the field quite a long time. I was curious how you came to be involved in the field of cell and gene therapy. Like, was that your training through school, or was that something you discovered after you had completed studies?
Yeah, that is a good question and maybe a unique history. I was trained in developmental biology and did my PhD on Drosophila,
Oh,
and I decided against moving to another place and like the environment. So I had to switch the field. And then I had the chance to start with the hematopoietic stem cell research nearby, and which is for sure very related to developmental questions we had before in the fruit fly. And one aspect was studying asymmetric cell division of hematopoietic stem and progenitor cells. And we searched for factors aggregating asymmetrically and ended up in identifying tetraspondents, and they were exosomal marker proteins. And that brought me in 2005, 2006, the first time into this knowledge that exosomes or extracellular vesicles exist. Yeah, and then I really appreciate it. And since 2009, we worked hands on, and yeah, it was a maybe an accident, a coincidence, and but a lucky move. And I really like the topic with the extracellular vesicles and the therapeutic potential.
right. And so then that was sort of the launching point to where your lab is now, right? Is that
Yes.
mainly what you are focusing on in your with your current research group? And maybe you could tell us some about your notable projects that you're working on right now.
Yeah, so one thing from hematopoietic stem cell research was that we used mesenchymal stem or stromal cells as stromal cells for hematopoiesis. And when we stepped into the field, we had to identify methods to characterize EVs. And by chance, we came across nanoparticle tracking analysis, and which we qualified and introduced with another group in 2011. And there we already described that there are mesenchymal stem cells secrete EVs. And then I was aware of the studies of Psyche Lim, who was one of the first to show that MSCs act via extracellular vesicles. Yeah, and so we had MSCs, we had the AVs, we had some methods to prepare them, even large scale. And then by a chance, in 2011, I discussed with the head of the bone marrow transplantation unit, Dietrich Behlen, about a GVHD patient, which was treatment refractory. And he considered as the last chance to treat her MSC therapy of Osiris. But that was the time when the phase 3 clinical trial became aware that it was failed. And so he discussed whether we may provide exosomes from MSCs, which was a huge challenge. We took it, and I think it was a quite successful application showing the power of the MSCVs. Yeah, and from then on, we focused on bringing them into the clinic. It was not GMP at these days. We took a lot of the challenges in the field. I think heterogeneity is a massive challenge. Some EV preparations are active, others are not. Yeah, and meanwhile, we have managed to immortalize them to produce clonal MSC cell lines and can conserve the therapeutic aspects of the EVs, which we explore with different collaboration partners in animal models. And now we think we have the prerequisites to proceed quickly into the clinics.
Well, that's good news. Okay, exciting news. That's so interesting about, so specifically EVs from MSCs. I mean, what's your one-to-one there? I mean, how many MSCs do you have to culture to get enough exosomes to treat a patient? Is that a rate limiting step?
Yeah, the dosing, that's a good question and an important question. And that was a challenge in 2011. What should be the dosing? And I think we learned a lot from the MSC field, where MSCs are administered in more than 1,000 clinical trials. And there was the average that about 1 million MSCs have been applied per kilogram body weight of the patients. And that was what we considered. And we allowed our media, ourselves, to condition the media for 48 hours. And so we reduced it to a factor of 2 and said treatment doses is the EV harvest of 500,000 cells per kilogram of the patient. And we applied them in this application in escalating doses. And we observed that already after the 30% dose, we saw an effect. And so I think the dosing was in the correct range.
Okay, okay, that's good to hear. So what is it about, like, provided you can produce enough MSCs to get enough exosomes to dose your patient, what do you think is like the special sauce in those MSC exosomes that is having the therapeutic benefit versus exosomes from any other cell type that you might be able to collect more easily?
So, yeah, that's a challenging question. I would not necessarily say MSCs are the best. Maybe there are other stem cells around or progenitors which produce very potent EVs. But if you are aware that MSCs are the second most transplanted cells after hematopoietic stem cells, and we have learned that MSC administration is apparently safe, at least in most of the studies, we would consider that also EV products of these cells would also be safe. And otherwise, because the administered MSCs will secrete EVs in vivo, we do not expect any side effects, and that's a good ration. So then there are good cultural conditions for MSCs. We again profit from the knowledge of the field. And if we would do it for, let's say, we start with neural stem cells, which we derive from iPSCs. Yeah, so what is the side effect of these cells? We don't know. I would not expect too many. But that was the driving force why we stick to MSCs. And I think it's the experience in the clinics and the easy handling of the cells. And these EV products are potent, but not necessarily saying that they are always the best for each indication. We discussed it in a dispatch article that there might be others as well.
Sure. And I can see that being actually a big touch point at the next ISCT meeting about whether you can almost continue to drill down into the exosomes, right? You mentioned it's sort of a nice trajectory or like to piggyback on the regulatory success of and safety of MSCs and then exosomes from MSCs, and then maybe even digging down further into isolating something from those exosomes that then you could just give to the patient, right, without any of the worry of transplanting whole cells into a patient, right?
Yeah, that is something we experience, that there are two ways to approach the EVs. On the one hand, there are many who see the advantage of cell therapeutics because the cells have a multimodular mechanism of action, so they regulate angiogenesis, the immune system, they work as stromal cells for certain stem cells, so they have certain target points. And therefore, there are good reasons for these therapeutic stem cell administrations, which is a side of hematopoietic stem cell transplantation, not cell replacement, very likely. So with that, we have now a stem cell product 2.0, which is cell-free, and it's very beneficial and less complex than the cells. But on the other hand, we have also to compare ourselves with those which work with small molecules, which have synthetic compounds which are defined, where you know where they bind to, which is the receptor, you know the mechanism of action.
Mm-hmm.
And yeah, with that, we have a very undefined product. But then we should say, if we want to organize a congress, we need to have money, we need to have speakers, we need to have the audience, we need to advertise. So we have different activities which we need to do. If we just have the venue or just have the money without speakers, it will not work. And I think a good therapeutic agent will press several of these buttons in an organism like the cells do.
Yeah.
But a mononuclear therapy will only have one activity. So maybe you have the speakers, but no venue. So it will not be a congress. And I think this is the charming thing of using cells or their products to play different aspects, which provides a lot of challenges because what's the concrete molecules? Yeah, there are plenty of, yeah, not
Right.
even one, plenty of. And which is the target site? Yeah, there are not only one, there are many. You have to approach the speakers, the audience, and so on. This is very comparable. Yeah.
Right, right. But then that just makes me think of the potential for probably more currently in the basic research side of things to identify ways to specialize, right, these cells, ways to, you know, prime them to go after a certain indication or therapeutic, right, disease state, pathology. I don't know. It just it starts. I should start the wheels churning. I think in a lot of people's minds about the possibilities that this type of therapy presents. But I agree. I like these conversations about, you know, whole cells versus just exosomes versus, you know, one specific small molecule and the pros and cons thereof. That would be a whole separate discussion. So maybe we'll
Sure.
have to have a part two to follow up on that.
Yeah, sure. Yeah, I started in stem cell research in 1999, so I observed the whole development, and I think there are great stem cell products, and so I believe that a stem cell therapy very often is better than a molecular therapy, and so the big challenge now with EVs is that they completely mimic. I would not say they are better, but the EVs are easier to handle. You can produce them in larger amounts. We can produce them from cell lines,
Right.
which you cannot. So they provide a lot of advantages, which make the product more market ready.
Mm.
Whereas cells might be only always expensive, and for a small patient cohort, I see chances that the stem cell product 2.0 cell-free can be scaled to much higher amounts and may reach much higher market readiness than any cell product. That is my thinking. Yeah.
Yeah. Oh, I like that logic. I think I'm curious how you would answer the maybe the regulatory challenge around, mechanism of action. If you have to provide a mechanism of action or a potency assay that's consistent, how would
Yeah,
you
this is somehow emerging as the $1 billion question. What is the mechanism of action? So I think there are different mechanism of action attributes in these products, which we also aware of MSCs. So apparently there is an impact on monocytes, there is an impact on neutrophils, there is an impact on angiogenesis. Several of these effects might be direct. There is evidence that there is also the complement system regulated. So we have multiple activities. And what we need to do now is we need to identify functional readouts where we can mimic some of these activities, which we do think play a role in vivo. So, for example, many work on monocyte assays. We have a multi-donor mixed lymphocyte reaction assay established, which shows the same activity in these assays. Then we observe in a graft versus host disease mouse model of the EV preparations. Some are active, some lack these activities. But the challenge is to also set up other assays for angiogenesis, for stem cell support, for direct impacts on neutrophils. And that is what we think we would need a function, a matrix. And this a matrix of different assays to monitor different activities in vitro. And yes, that's a very huge challenge. And so to say, I would say for the moment, if you show that your product can be produced reproducibly and you have metric characteristics that are fulfilled,
right?
maybe for now we need to show they are potent in animal model and have one or two assays in addition, functional assays. And over the next few years, we need to develop potency assays. But I think hardly any group has potency assays above, maybe monocyte macrophage modulatory assays.
Right. No, clearly a lot that still needs to be worked out. I'm glad to hear you're working on it and thinking about it. I imagine your group is going to bring a lot of posters to Paris. Are you
I
preparing to
hope
come?
so.
Yeah. Have you booked your train ticket yet? You're just down the road.
Yeah, it's for you Americans, it's immediately around the corner. So I live in Cologne. This is maybe a 4 hours car trip. Yeah, and with a train, there are speed trains. You may be there even quicker. So that is, it's quite easy to go there. So compared to the States, the area of Europe is pretty small, and we can reach almost each places quite quickly by plane or by train.
Yeah. Oh, that's fantastic. I mean, I was curious to get your thoughts, in case, for a lot of early stage professionals, this might be their first international meeting. Do you have any advice for them, how they should take advantage of it?
Yeah, on the one hand, it's of course the exciting program of ISCT. I think there are some roundtable discussions which will be great. So I learned that it's organized in different sections, early stage and early clinical projects, and then later clinical projects. I think it's nicely structured that we can have the different levels of the of how to bring products into the clinics. We are discussing that there will be an educational session on the last day about EVs, about the current challenges. So I think it's an exciting program, but one advice could be that at least for the people coming from abroad, maybe they should spend some extra days because Paris is really worth a visit. So there's a lot of cultural things to do. It's an exciting city. It's good night clubbing, cultural aspects. So it's not, you're not in the boring desert. You're in a really big European metropolitan city, which is worth a visit together with ISCT. Don't do the one or exclusively the other. Join both sides and bring enough time.
Yes. All right. You heard it here first, guys. Do whatever you have to do to extend your trip over there. Take a couple extra vacation days and enjoy it. Well, that is what
Yeah,
I'm planning to do. So I am very
yeah,
excited to
excellent.
hopefully, yeah, hopefully we can connect in person there and hopefully we'll see a lot of other people there in May. So, but yeah,
Yeah.
again, Bernd, thank you so much for taking so much time to chat with me. I think we reached the end of our time. So I wanted just to thank you for sharing so much about your research and just helping us start this series off in 2023 with a phenomenal conversation and lots to think about coming out of this chat. So thank you.
Yeah, thank you very much, Ashley. I appreciate it very much.
All right, you take care. Bye.
Okay, take care. Bye.